Welcome to the first video in the biology module lecture series. In this video, we're going to go into the cell, or as it's commonly called, the basic unit of life. Now take note that throughout this unit, we're going to have general overviews, but there is more material and there's more detail than what we can cover in these video series. If you're unsure about some material, just refer back to your old textbooks from your previous biology courses. Anyways, let's begin. Starting off with the cell theory. The cell theory is basically four tenets that are an overview of what cells are and how they pertain to biological life. The first tenet is that all living things are made of cells. The second is that the cell is the basic unit of life. Third, cells have to arise from pre-existing cells. And last, cells carry genetic information as DNA, which is passed on from parent to daughter cells. Take note that viruses are not considered living because they are not comprised of cells, and they also cannot reproduce without help from a host cell. With that covered, let's go into eukaryotic cells, which will be the more prominent cell that's covered on the MCAT. Below is a picture of a generalized animal cell. Take note that a eukaryote is designated by having membrane-bound organelles and a nucleus. Remember that the membranes of eukaryotes are made by phospholipids, which are amphoteric cells that create hydrophobic and hydrophilic areas. The first organ, and the one that eukaryotes are generally characterized by, is the nucleus. The nucleus carries genetic information in the form of chromosomes and is organized into genes. The nucleolus is a subsection where the rRNA is synthesized. The next bit is the lysosome. These have hydrolytic enzymes, and they break down molecules that are ingested and cellular wastes as well. The endoplasmic reticulum is divided into rough and smooth. The rough endoplasmic reticulum can be characterized because it's studded with our ribosomes, and it translates proteins. The smooth because there is no such studding. It's used for lipid synthesis and detoxification. Moving down the chain, we go into the Golgi apparatus. Think of this as a warehouse that ships materials as it gets in. Cell products from the endoplasmic reticulum or ribosomes in the cytosol are modified, packaged, and shipped to specific locations from the Golgi apparatus. The last thing we're going to go into is the peroxisome. This contains hydrogen peroxide, and it breaks down long fatty acid chains. As you would suggest, it participates in phospholipid synthesis and the pentose phosphate pathway. One organelle that we're going to go into in greater detail is the mitochondria, because it is commonly tested and the site of cellular respiration. Because of this, it's characterized as the powerhouse of the cell, and it's unique in the sense that it undergoes binary fission independent of the nucleus and carries its own genetic information. This leads a lot of biologists to suggest that mitochondria were bacteria that were consumed by a larger bacteria and self-sustained within the larger bacteria. It generates ATP for the use in cells, and it's often more common in cells with higher energy function. So, for example, muscles. Mitochondria generate ATP using cellular respiration, which is a combination of glycolysis in the cytosol, the Krebs cycle, and the electron transport chain, ATP synthase being the final step, which converts ADP to ATP. As you would suggest, using oxygen in the Krebs cycle generates CO2, which the cell has to find some means of removal. If you would like a more in-depth look at cellular respiration and the Krebs cycle, again, just refer back to your old biology textbooks. It's in your best interest to have this process down, and although photosynthesis is roughly tested on the MCAT, it's not going to be put into as great a detail as cellular respiration. Now that we've talked about the organelles, let's go into the cytoskeleton. The cytoskeleton, as you would guess, provides stability structure, and it also transports things across the cell. It's comprised of three components, starting with microfilaments, which are actin. They provide structural protection and muscular contractions via myosin. These will be more common in muscle cells because of this reason. The second criteria, or the second category, are microtubules. They are made of tubulin, and they create pathways for mortar proteins, with kinesin moving away and dyneins moving towards. They contribute to cell motion. Also, during mitosis, they contribute to the process by making centrioles and centrosomes. Intermediate filaments are what connects cells together in an adhesive process, and they maintain the cytoskeleton as well. Also, they keep organelles in place when they're supposed to. A couple other tissues we'll go into are epithelial and connective. Epithelial, for more complex organisms, is more or less classified as your skin. They cover the body and line the cavities, and they protect against pathogenic invasions, along with, in some organisms, controlling and regulating body temperature. The cell types are classified as simple stratified or pseudostratified, and they're either cuboidal, columnar, or squamous in shape. Connective tissue, as the name suggests, support the body and provide frameworks for epithelial tissues. So what that means is that the epithelial tissue will be on the topmost layer, followed by connective tissue attaching it to the rest of the body. Now that we've talked about eukaryotes, let's talk about prokaryotes. And with prokaryotes, since they are generally pretty simple in structure, they're often classified in different ways. Prokaryotes are noted because they do not carry membrane-bound organelles and do not have a nucleus. Their genetic material is a single circular model of DNA, and it looks something like this. They also undergo binary fission and can have plasmids introduced to them. If you want to classify them by shape, there is spherical, rod, and spiral. Spherical being cosi or coki, rod being bacilli, spiral being spirally. In general, the way I remember this is spirally is a spiral and a bacilli is a bacteria, which I often think of as a rod. So that means that cosi has to be spherical. You will be tested on these classifications, so commit them to memory. Other ways to classify prokaryotes include metabolic process, and there's 4 major ones based on whether they use oxygen or not. Obligate aerobes, as the name would suggest, are obliged to use oxygen. Obligate aerobe. Obligate anaerobes are forced to use non-oxygenated processes. As such, oxygen is toxic to them, so they can't survive in these environments. Facultative anaerobes switch back and forth based on the environment around them, and aerotolerant anaerobes cannot use oxygen but can survive in oxygenated environments, unlike obligate anaerobes. Remember that these are for prokaryotes only, so if we're talking about eukaryotic cells, you can dismiss these classifications. They will try to trick you on test day with this, so be aware of that. One methodology that will commonly be tested on the MCAT is the Gram stain. You need to know what a gram-positive and a gram-negative is going to look like. A positive for the reaction is going to turn the bacteria purple. This indicates the presence of cell walls containing peptidoglycan and lyoteichoic acid. On the bottom left is a picture of what this color is going to look like. Gram-negative, on the other hand, has no such color change. You'll see instead a slight pink. This indicates thin walls and an outer membrane of phospholipids and lipopolysaccharides. Now, you're probably thinking to yourself, why would we bother with a Gram stain? Well, it's to show whether or not there's a cell wall or not, and this is to classify cells accordingly. So now that we've talked about some classifications of prokaryotes, let's go into the genetics and the growth of prokaryotes. As stated earlier in the lecture, prokaryotes are comprised of circular DNA, with plasmids that can be introduced. There are 4 methodologies for genetic recombination in bacteria, and it's a very common testing area to be talking about such things as genetic engineering and augmentations of these said bacterial genetic materials. Starting first, let's go into transformation. This is the acquisition and assimilation of genetic material from the environment, i.e., plasmid vectors incorporated into the DNA. This is commonly used when biochemists want a specific type of bacteria. The next is conjugation, which is the transfer of genetic material from bacteria to bacteria via a bridge. If a bacteria has an advantageous trait or gene, it will often share it with those around it. Transduction is the transfer of genetic material via a bacteriophage as a vector. In genetic recombinations in the laboratory, biochemists will often put this into a virus and infect bacteria to propagate a change, and then they will select via chemicals or other methodologies the bacteria that have successfully incorporated this vector. Lastly, there are transposons. Transposons are genetic elements that insert or remove from the genome. Bacterial growth follows a pattern, and this is true for all bacteria. The lag phase is when the bacteria is adapting to the new conditions around it. There often isn't any growth seen in this phase, meaning that there's no fission reactions, because the bacteria still has not acclimated. So from a cellular standpoint, it's not sure if reproducing is a good idea. The next is the exponential phase. This happens after the bacteria has successfully adapted to its new conditions. You're often going to see it explode in growth, and the amount of bacteria in this environment is going to increase exponentially. The stationary phase is when the resources in the environment equal the amount of bacteria present. It's going to create a plateau, which you can see in the graph below. And lastly, the death phase. This is when resources are insufficient, so the number of cells is going to decline, often very sharply. We've covered cell life. Now let's go into viruses and subviral particles, or pseudo-living particles. Starting off with what a virus is. Viruses are genetic material that's enclosed in a capsid that infects and utilizes host cells to reproduce. Note that this can be DNA or RNA in terms of retroviruses, which go against the central dogma of biology stating that DNA goes to RNA to proteins. Instead, retroviruses go backwards using reverse transcriptase to get DNA from RNA. The DNA that's synthesized from this enzyme is then incorporated into the host's genome. Often, bacteriophages are the viruses that you will see on test day, since they are the vessels that are used as vectors in genetic engineering. Speaking of bacteriophages, they have two separate kinds of life cycles. The first being the lytic cycle. This produces virus until the host cell lyses. It's a very hostile takeover, and the cell's machinery is taken over and forced to produce virus until the host cell bursts. The second is a little bit more benign. It's the lysogenic cycle. Viruses in this cycle will integrate into the host and reproduce along with the cell. So it's basically a hitchhiker that follows the cell as it reproduces and as it lives its life. Take note that if the conditions are right, a lysogenic bacteriophage will enter the lytic cycle and thus cause the cell to be controlled and burst eventually. Take note that if a cell has a lysogenic bacteriophage in it, sometimes that virus is going to occasionally produce other bacteriophages, but not at the expense of killing its host. Of these two, obviously the lysogenic cycle is a little bit more advantageous because it keeps the host alive. As a parasitic pseudo-living organism, viruses will generally prefer not to have to search for hosts, although technically they're not living. But as you would suggest, it's in their advantage to keep their hosts alive. The last thing we'll go into are subviral particles. The first being prions. These are proteins that infect other cells and trigger misfolding of other proteins in the cytosol. Viroids are plant pathogens that are small circles of complementary RNA. As you would suggest, these complementary RNAs will turn off genes and alter cell functions within the nucleus. So that'll cover our introduction to biology. Cell theory is a very commonly tested subject on the MCAT, so make sure you have a good handle on this come test day. Please join us in the next lecture as we go into reproduction.